I'm excited to introduce you to a new drug to treat COVID-19. The drug is called Molnupiravir, and it works as a nucleoside analog. You can see the chemical structure of Molnupiravir here, and Molnupiravir can be turned into MTP, where it has 3 phosphates on it, and in this form, Molnupiravir can be incorporated into the viral RNA as the virus is attempting to replicate inside of cells. This leads to extensive viral mutations such that the virus is no longer viable. This image is just showing an overview of SARS-CoV-2 replication, which you've seen before. So let me just review briefly. Here's the SARS-CoV-2 virion binding to the ACE2 receptor and entering the cell. Once it gets into the cell, the genome is released. Remember, the genome is RNA. The RNA starts being translated, and one of the really important proteins that is translated is an RNA-dependent RNA polymerase, shown by this complex of proteins here. The RNA-dependent RNA polymerase will copy that viral RNA. This is critical for making the negative sense RNA and then the positive sense RNA that will be packaged into the new virions. Molnupiravir is going to mess up this part here, the RNA replication part. Let me explain how that works. Here is the RNA-dependent RNA polymerase. Here is the genomic plus strand RNA that came out of the virion that entered the cell. And this is the new RNA that's being replicated by the RNA-dependent RNA polymerase. Normally, the RNA-dependent RNA polymerase would be building this new RNA out of A, C, G, and U nucleotides. But when the drug is present, shown by these yellow M's, the Molnupiravir can be incorporated in the place of C's or U's. And when this happens, you have these M's inserted all along the genome of the virus, and this is messing up the code in the virus. This now leads to mutagenesis, where that negative sense RNA with all the M's incorporated, when it now gets copied into the positive sense RNA that will be packaged in new virions, it has the incorrect nucleotides in its sequence. So the more the SARS-CoV-2 virus tries to replicate, the more messed up the RNA becomes. The mutagenesis in the RNA is so severe that the SARS-CoV-2 virions are no longer able to function, and the infection is cut short.